Hi everyone, thank you all for attending my talk. My name is Evan, and I work in Mert Sabuncu's lab. This work was done in collaboration with Adrian. In this presentation, we'll introduce a method to learn conditional deformable shape template for brain anatomy. An atlas, or template, provides a standardized set of coordinates to determine specific sites within the brain. In this presentation, I will be using label atlases as shown by the example below. It assigns label probability at each voxel of the brain. There are a lot of applications for brain atlases. For example, we can register a subject to a common template for a standardized study, or we can identify brain structural changes over time. There are even methods that use brain template for segmentation. Unfortunately, there are downsides of using a fixed template. We all know how heterogeneous our population is. Demographic, clinical, or other confounding factors can influence the shape and size of the brain regions. As a result, a single and fixed template can struggle to accommodate complex structural differences across a heterogeneous group of individuals. In this work, we introduce a method to deform templates in order to capture attribute-specific variation. The deformation is an explicit function of a given attribute vector. We present an end-to-end learning strategy to train the proposed neural network and present empirical results that demonstrate the utility and reveal interesting neuroanatomical shape variability associated with age, sex, and Alzheimer's disease. Here's the setup. As I mentioned before, we have a probabilistic template that assigns probability of a given label at each voxel of the volume. In other words, a categorical distribution at each voxel. We are interested in modifying the template or prior as a function of demographic and clinical variable, all of which are collected in an attribute vector. Thus, our objective is to learn conditional distribution of a given set of attribute and template. This idea can be captured by the following objective function. In this setup, Z is the latent variable that parameterizes the attribute-specific deformation of the template. For the sake of simplicity and implementation, we use a multivariate Gaussian with diagonal covariance as the distribution of the latent space. For the likelihood term, we assume that is generated by warping the template with the deformation field that is created from the latent variable. The objective function is in general intractable. A simple way to optimize this objective is to do Monte Carlo sampling. We first rewrite equation 2 as an expectation. We can then take sample of P of Z given A and take the average. We use a neural network for different parts of our model as shown in this figure. Attributes are passed to a fully connected network to produce a mean and variance of the multivariate Gaussian. We sample a Z from the Gaussian distribution, which is then upsampled using striated convolution. The last convolution layer outputs a velocity field. The reason that we are outputting a velocity field instead of a displacement field directly is that we want to constrain the displacement field to be smooth and continuous mapping with derivatives that are invertible. In other words, the diffeomorphic constraint. We don't want our output deformation to, for example, fold the brain in an unrealistic manner. By outputting a velocity field and then integrating it back to the displacement field, we can enforce that constraint. Finally, we use the integrated displacement field to deform the template. To evaluate our algorithm, we use 415 subjects, 255 females and 116 males, with ages ranging from 18 to 96 years old. The dataset includes 100 subjects diagnosed with Alzheimer's disease. We randomly pick 375 subjects for training and the remaining 40 reserved for testing. We want to evaluate how well our algorithm is able to deform the template given a set of attributes, namely sex, age, and diagnosis of Alzheimer's. We experimented with two templates. For the first template, called template 1, we used the one-hotted encode segmentation from an independent healthy subject in the MCIC dataset. The second template, we use a probabilistic template from the Buckner dataset. Since we have two different templates, we have two different models. Model 1 uses template 1, and Model 2 uses template 2. Here is the qualitative experiment. We show the coronal and axial view of the model trained with probabilistic template. We deform the template to capture the look of a 20 and 70-year-old healthy female and a 70-year-old female with Alzheimer's disease. Difference map between the pairs of deformed templates are shown in the last column. An obvious change is the ventricle, shown in green. As we get older, the ventricle size increases. Similarly, if we have Alzheimer's, the ventricle also gets bigger. Here's a short movie showing how our model 2 deformed the template as we increase the age attribute. We can have a more quantitative look by measuring the volume of the deformed template as a function of age. The solid line are for healthy males and females, whereas the dotted line are for Alzheimer's male and female. We can see that as we age, both the white matter and gray matter decreases over time. In addition, in the presence of Alzheimer's, the volume is even smaller. For this experiment, we used the attribute of each subject, deformed the template, and compared the deformed template to the actual segmentation label of each patient by comparing the DICE score. Do not think of this as the segmentation task, but rather how well a deformed template is able to account to the attribute of each subject. Remember, during training, the only thing that this model sees is the attribute and the fixed template. From the table, we can see that if we use a fixed template from a random subject or a probabilistic atlas, we get a low DICE score because the fixed template cannot account for the subtle anatomical difference across a heterogeneous group. On the other hand, our model is able to deform both the template to capture these subtleties. The box plots show the same experiment but broken into different groups of interest. Finally, we can use our model to do shape analysis by outputting the deformation given an attribute. On the left figure, we computed the surface distance between a 90-year-old healthy male hippocampal surface and a 18-year-old male hippocampal surface. On the right figure, we did the same thing, but we computed the sign distance between the surface of a 65 healthy male and a 65 male with Alzheimer's. The color bars show the sign distance, red for positive and blue for negatives, and the units are in millimeter. Thank you all for listening.